So Andrew's a fifth-year PhD student in the lab of Jean-François Prin, and he studies the mechanisms of mitochondrial dysfunction in neurodegenerative diseases. And in his research, Andrew uses mass spec-based techniques to characterize a mitochondrial peptidase that's implicated in cerebellar ataxias. So in terms of proteomics, Andrew has provided mass spec analysis for numerous projects, both within and outside of his lab, that involve proximity labeling, relative quantification, protein turnover measurements, and more. So with that, I'll just turn it over to you, Andrew.
Thank you, Lucia. This is my first recorded meeting, so I'll be very careful with my words. Okay, so yeah, today we're just going to jump into and hopefully demystify some of the scariness surrounding mass spec. And I hope that I'll be able to convey some of the principles clearly, and that by the end of it, everybody in this meeting will have some cool experimental ideas of how they might be able to apply mass spec within their own projects, because it is a very powerful technique that can seem daunting at first. First, just the broadest question of all, which is why to perform mass spectrometry. And there can be a number of answers to this one, simply to determine the precise molecular composition of a protein, to identify new proteins or metabolites, to gain structural information on proteins or a group of proteins, to quantify the abundance of a protein in different samples. And then finally, the most powerful part of it all is to quantify global changes in protein abundance in an unbiased way. And you can do this, you can look at abundance, modifications, or interactions. And when you're profiling everything all at once, of course, it takes on the omics term. So this is what is called proteomics. And I'll go through some things now, and hopefully by the end, everybody will be obsessed with proteomics like I am. Okay, so first, a really basic scheme for the components of a mass spec. We'll just look in blue here first. Basically, you're going to feed a liquid into the mass spec, and in this liquid, you're going to have proteins or peptides. And then the first thing that is in all mass specs is an ionization source, because we are loading a liquid and we want to get it into the gas phase, and we want to ionize our peptides or proteins, and then desolvate these ions, and then they will travel in the gas phase through these other components. And these other components in the mass spec are what's called a mass analyzer. And this is either a quadrupole or a hexapole. So basically a series of 4 or 8 rods that have different currents applied over them. And this is able to separate these ions by their mass to charge. Because I do need to say that in the ionization source, at least for all of the proteomics workflows and things that I'm going to be talking about today, your peptides or proteins are going to gain positive charges. And so for the source that I'm talking about, they will gain many positive charges, and it's a bit of a random process. But what the mass spec sees is not the mass; it will always be the mass over the charge of the ions of interest. And so the mass analyzer here is able to filter out certain ions of a mass to charge through some neat physics that I don't quite understand. But after you filter out certain ions of interest, then they will go to the detector, and then you'll get an idea of their precise mass to charge and their abundance. And just to say that these two portions are under a high vacuum, because when you have ions in the gas phase, you really don't want them to collide with any molecules that are potentially in the air. So that's the basic workflow of the simplest mass spec. And one thing that I have kept hidden from you guys is that I told you that we feed in a liquid of interest, but one really critical part to all of this is to have an HPLC. So this is a high pressure liquid chromatography upstream of the mass spec. And what this does is it allows you to separate proteins and or peptides by hydrophobicity by doing reverse phase chromatography. And so this, while you can inject directly into the mass spec, if you have complex mixtures or if you have certain salts or buffers and things like this, it's really, really, really nice to run them on an HPLC first, and then you run them on a C4 or a C18 column. Hydrophobic column, the C4 or C18 is the length of the alkyl chain. And then you have some temporal resolution for what's actually going into the mass spec. So it's not like you're throwing everything into the mass spec all at once and it is unable to sort of pick out everything at once, because there are some limitations. Okay, we're going to zoom in a little bit now on the ionization source to the one that I am most familiar with, because there are many different kinds of ionization sources in mass spec. But the one that I am most familiar with, and the one that is always used in proteomics, is something called electrospray ionization. And so what's happening here is that you can imagine that the HPLC is upstream here, and you are injecting a liquid into this Taylor cone here, which is basically just a cone that has a voltage applied across it, and operates at high temperature. And so in the Taylor cone, now you have the liquid that is going into a highly charged aerosol. And from this aerosol now, these ions will desolvate, meaning they will enter the gas phase completely. And then these gas phase ions are the ones that will go into the capillary here, and this is applied under vacuum. And then you can imagine that the mass analyzer will be downstream of here, and then the detector will be downstream of here. One thing I want to say about ionization is that for ESI, it's something called a soft ionization technique. And this is really important for doing protein or peptide work, because what that means is that the protein or the peptide will not fragment or break apart in this spray formation. There are some techniques, especially if you're doing small molecule mass spec, you can use a harder ionization technique like electron ionization or something like this. But for proteins, if we want to look at and eventually figure out the very precise mass of something, we don't want it to fragment right when it's getting ionized. So with electrospray ionization, it's really nice because it's a soft ionization technique and it generates multiply charged ions. And this is different from other soft ionization techniques. But this is really good for proteins and peptides because it widens the mass to charge range. Because you can imagine if you have something massive, and in a technique like MALDI, which is it shoots a laser at a solid matrix to create ions, that'll only generate singly charged ions. But if you have something massive, since the mass spec is measuring the mass over the charge, it's beneficial to have something that's highly charged, because then your mass to charge will fall into a window that actually is visible to the mass spec. I think that's all I want to say there. I'm going to show you guys a very cool picture of my favorite room in the McIntyre building, which is our mass spec room. And so just to give you guys sort of a real life visualization to that schematic earlier, this is our HPLC here, which has a C4 or a C18 column to separate peptides or proteins by hydrophobicity. And then it's not connected, but this line here that's going into the waste, you plug it into the mass spec here. This is the ESI source, and then there's the mass analyzers and things are sort of here where my cursor is. And this is what's called a QTOF. And so Q means quadrupole mass analyzer, as I described before. So that's going to be somewhere in here. And then TOF is unique to, this instrument, which is called time of flight. And if you look back here at this navy blue sort of very tall rectangular chamber, this is the time of flight chamber. And what happens here is the ions are coming in after being filtered by the mass analyzer. And then there is a 90-degree electrical pulse that then pushes the ions, and they travel in this parabola. And then depending on their mass to charge, they will either travel faster or slower through the parabola. So you get separation there, and then they hit the detector at the bottom here. There's another really common kind of mass spec, which is called an orbitrap, that I won't get into, but the orbitrap is a little bit higher sensitivity than the QTOF, and that's what will be used if you send out samples to a proteomics facility or something like that. But the premise is pretty much the basic components that I outlined earlier are still the same. Just two little nomenclature things. The resolution of a mass spec is basically the ability for the mass spec to separate two peaks of a very similar mass to charge. And so resolution, you want a higher number. And then mass accuracy here, you want a lower number, and this is shown in parts per million. And what this means is that for even something up to 1,000 Daltons or 1,000 mass to charge or 10,000 mass to charge, the mass accuracy should be within a fraction of a Dalton. So this is a really, really, really sensitive technique when your instrument is working properly. Okay, so now I want to show you guys what the data looks like just in a really simple form. After it hits the detector, it gets digitized and goes to your computer. And so this is a single protein injected on the mass spec. This is myoglobin here. And the first thing that you'll notice is that looking at what's called the charge distribution envelope, I told you earlier that with ESI and with proteins, you're going to have multiply charged species, and they're going to be positively charged here. And so a protein, in this case, we are injecting the protein in formic acid, which acts as a proton source. And so in the during the ionization process, proteins, basically any free electron pairs on proteins have the ability to go and grab protons during the ionization process. And so in proteins, it's going to be your basic amino acids, so like lysine and arginine, and to a lower extent histidine, and your free N-terminus. And so you can imagine that for a really large protein, you have a lot of basic amino acids, and this process is kind of random. So you're going to get a distribution of highly charged species. Yes, and just one thing to hammer home is that the mass spec is seeing the mass to charge here. And so you can notice that at lower mass to charges, you are having the more positively charged ions because it's M over Z. And so even from a single ion here, so if we take the 15 plus ion, we can back calculate the intact mass of the protein that we injected just by multiplying it by 15, because that to M over Z, multiply it by Z. And then you subtract the 15 protons that it accumulated during the ionization process. And you can see here that from the data, we are in 0.3 Daltons off from this simple calculation. So now I talked to you guys a bit about resolution. If you have a really powerful mass spec that has a high resolution, you can zoom in on the 15 plus ion here, and then you can zoom in again. And it's a bit of a fun thought experiment. It's not necessary to really, process data and things like that. But if you understand what the data looks like, then it's always easier to sort of go back and validate what the softwares are spitting out at you. And so when you zoom in on that 15 plus peak, now we get into the concept of isotopes in mass spec analysis. And so for the sake of simplicity, we're only going to focus on carbon 13s, which are the most, they have a high natural abundance, and there are carbons everywhere in proteins. And so really the mass spec only cares about carbon 13 and maybe nitrogen 15 to some extent. But why is this important is because if the resolution is high enough and you zoom into a single ion, you can actually see an isotopic distribution. And so this is a smaller protein now. We have angiotensin. And if you notice, we have the monoisotopic mass, which means all carbon 12. And then you have these peaks that are separated by 1 Dalton. And so this is basically 1 carbon 13 in the protein. And then if you keep going, it's 2 carbon 13s. And the larger the protein, the higher the probability will be that you have multiple carbon 13 atoms. And so you will tend to not see this monoisotopic mass peak in larger proteins. Angiotensin is pretty small. It's only 1,000 Daltons. And so if we go back now and zoom in on that 15 plus ion from myoglobin, which is 16 kilodaltons, so about 10-fold larger, you can see that the monoisotopic peak, you don't really see it. And now you have this distribution of isotopic peaks. And the only other confusing thing is that we were looking at the singly charged the +1 ion here. And now at +15, the spacing between the peaks is not going to be 1 Dalton or 1 mass to charge, but it's going to be 1 Dalton divided by 15. And so it's, you guys don't need to know this, but this was something that sort of confused me for a very, very long time. And when I finally understood it, it helped me to sort of see really how powerful the mass spec is. Okay, now we'll get into some practical considerations, and I'll show you hopefully a lot of examples that might inspire you. So to focus on the easiest example first, we're going to just look at intact protein mass spec analysis. And so this is not proteomics yet, but this is just, for instance, we have one protein we want to, see its precise mass, see if there's any modifications. And so, excuse me, the considerations for this, you really don't need much sample at all. For most proteins, you can get away with injecting 500 nanograms or one microgram on the mass spec. Certain proteins don't ionize well. And so we have examples of proteins that we know are fine, but in very rare cases, they just don't show up on our mass spec. And so it's not 100% perfect all the time, but with some tweaking, you can see pretty much all proteins. Okay, so you need, let's say, 1 microgram of a protein per injection. What you're going to do is you're going to acidify it, because I said before, in positive mode, which is how we're going to do all of this stuff, we want a proton source for the electrospray. And for a protein, we'll inject it onto a C4 column, hydrophobic column, elute with acetonitrile. And this is just a mobile phase to elute stuff off of a hydrophobic column. You can use methanol if you want it as well, but acetonitrile is the most common. And so the length of the gradient for a single protein, it doesn't really need to be anything super long, because we're not trying to separate a complex mixture. We just want to clear out some of the salts at the beginning and then inject it onto the mass spec. So we do this in 10 minutes. And here, this is compatible with many buffers. And since we're running it on an HPLC first, we can have salts and things in the mix. But if you were doing a direct injection into the mass spec, which is common, then you need to be a bit more careful. But for all of our stuff, we have salt reducing agents, and it's fine because we run it on the C4 first and we don't inject it right away. Detergents are not so good though, so you want to remove those before if you can. And I'll show you now some of the exciting applications for a technique like this. And what's really, really striking about this kind of workflow is that you don't need much protein at all, and you can get data really, really fast. And so for one protein, it takes us about 30 minutes from top to bottom. And just to say, on the HPLC, there is an auto sampler, so you can just queue up a bunch of your samples and leave for the night or leave for 30 minutes if you want. Okay, so for an intact protein run, I'm just going to show you guys a bit what the data actually looks like on our software. And so up at the top here, this is not a mass spectrum, but this is a total ion chromatogram. And so on the x-axis, we have time, and on the y-axis, we have the intensity. And basically, you can just think of this as the total intensity of the ions at a given time. So let's say that this is a single protein here coming off of the column at a given acetonitrile percentage. If we had another smaller protein, we might see, and we might see another peak here earlier or something like this. But we only injected one protein here. And so what we're going to do is we're going to take the total ion chromatogram, which is the intensity of all ions at a given time. And then we're going to average the spectra across the peak to see what ions are composing this TIC peak here. And then this is a mass spectrum that is composing this peak. And if you look, it looks similar to the myoglobin example before. It's just a little bit compressed. So these are all of these multiply charged peaks that the protein has adopted. And from this, we can do a deconvolution in the software to basically back calculate the mass of the intact protein from all of its charged species. And we did the example before where we just took one, like we just took the 15 plus ion and we back calculated. But deconvolution will take into account the entire charged state envelope. So it'll take into account all of the charged ions and then give you a really, really precise mass. And so in our lab, we work with a lot of recombinant proteins. And so this was a mutant of this pink 1 kinase. And you can see that the predicted mass was here. And you can see that we were a fraction of a Dalton off. And even at 50 kilodalton, it's still very, very accurate. So this was a validation that the correct mutation had been integrated into the protein that we purified. Another example that led to some serendipitous findings came from a collaborator who wanted to inject synuclein, SNCA, protein into mice. And he was making it recombinantly, but for anybody in the call, if you're buying recombinant proteins from places, it can always be good to do quality control checks, and it doesn't take much and it's super fast. And so we were receiving synuclein from somebody in the Ted Fawn lab. And when I ran it on the mass spec, I noticed that the predicted mass for acetylated synuclein was correct, but there was this mystery peak at 183 Daltons that was about 20% of the total product. And I had never seen this mass shift before, but after some Googling, we found out that this was actually a covalently modified protein from a certain kind of protease inhibitor from AEBSF. And so AEBSF, the fluorine is capable of leaving, and then you're left with AEBS conjugated protein. And so this was a really important step to do before injecting into mice. And when from Ted's lab altered the purification protocol and used a different protease inhibitor, and we got rid of that peak. And so that was really nice. And then one of the other benefits is that you can also see degradation products. And you can see here, they're very, very minor, but when you zoom in, there are peaks whose masses correspond almost identically to truncation products. And so we had some N-terminal truncations here, which are very common in E. coli recombinant protein production. And so you're able to minimize a lot of these things when you have mass spec as a readout, because some of these things can be difficult to see on a gel. Okay, so beyond quality control, you can also monitor reactions and enzyme kinetics using mass spec. And this is a quick example from my work where I'm studying a mitochondrial peptidase that cleaves targeting signals. And so we are able to order synthetic peptides. We're looking at TICs here, but the same premise sort of applies everywhere. So this is the full length peptide 1 to 24. When I add in my enzyme of interest, we see that there is a specific product formed, shown in red here. And then when I add more enzyme, it goes to completion. And if you set up your reaction right to match the Michaelis-Menten parameters, you can actually get some KMs and KCATs from these kinds of setups. And so I was able to get the KM for the cleavage of this peptide using mass spec. It is not very high throughput because each one of these dots, and if you're doing it in replicates, can take somewhere 30 minutes to an hour with washes in between, but it's not just for quality control. You can monitor reactions as well. Here's another quick example where we were able to gain some structural information using mass spec, and this was on my recombinant protein here, and it's a heterodimer, so we have MBP alpha and MBP beta, both whose masses made sense. And then there's a technique in structural biology called limited proteolysis, in which you incubate your protein of interest with a peptidase in very dilute amounts. So we're doing a 1 in 1,000 ratio here. And the idea is that if there are mobile parts that are outside of the protein fold or that are just sort of flopping around and accessible to these peptidases, that they will get cleaved off. And if they're unstructured or if they're in loops or things like this, it can be very handy for crystallization or also to gain structural information on which parts of your proteins might be unstructured. And so if we just focus on this panel here, upon addition and incubation with chymotrypsin, which cleaves after hydrophobic amino acids, there was a really specific product formed here, which was a truncation of this free predicted C-terminus in MPP alpha. And unfortunately, it did not crystallize, which was one of the goals, but it did provide us with some information to validate the alpha fold structure here and suggest that this C-terminus really is free and accessible. Okay, so one last example for intact protein. Shafkat in my lab works on recombinantly producing a kinase. And after purifying the kinase and running it on a gel, everything looks great. It looks very, very, very pure. But the beauty of the mass spec is that this one bend on an STS page can actually be composed of many different species that are not known to you. And so what Shafkat saw was that his recombinant protein actually was multiply phosphorylated. And you can see that basically each of these peaks are separated by 80 Daltons, which corresponds to a phosphate. And this was a big problem for crystallization because in structural biology, you want a really homogeneous preparation. And the way that mass spec informed us sort of propelled Shafkat to create this new setup in which he was co-expressing his protein with a phosphatase. And this was very, very, very cool because now after co-expression with the phosphatase in E. coli and purification, he went from having this multiply charged protein to a zero phospho protein. And then he could control the reaction by, adding ATP and then getting it to a singly phosphorylated form. So mass spec was very, very helpful here. And now, of course, this is all great. But the big question here that will let us pivot into proteomics is, okay, we know that it is phosphorylated based on the specific mass to charge shown here, but how do we find out where a post-translational modification might be in a protein? Because everything I've told you thus far has been sort of not site specific or sort of resolved in the amino acid sequence. And so now we're going to talk a bit about Tandem MSM. And I lied to you guys at the very beginning because I showed you a very simple picture of the mass spec, which was an ionization source, mass analyzer, detector, and then you get your ions in some abundance. Now, I left out that after the first mass analyzer, there's something called a collision cell or an activation cell. And what this does is that you can now take your parent ions, is what they're called, and then in this cell, you bombard them with a neutral gas, and then you can actually fragment these ions. So let's now think about them as peptides, right? So you have peptides that are ionized. You have an intact peptide here. You can let it go and hit the detector, and then you know what the mass to charge is of that peptide. But two peptides can have the same mass to charge, right? Two peptides of different sequences can have the same mass. But now with this collision cell, you bombard with a neutral gas, and then this randomly fragments the peptide. And from that, you can go and identify the identity of that specific peptide and then associate it with the MS1 level identification that you got initially. Okay, so that is the basics of Tandem MSM. And I just told you that, yeah, you're going to inject peptides, but when we are injecting peptides, and especially complex mixtures, there is a very standard workflow in all proteomics experiments. And so I'm going to take us now from a mixture of complex proteins, and then how do we get to good proteomics data? And so we take proteins, and then the first thing that we're going to do is we're going to denature them. In urea buffer is super common. Then we reduce the disulfide bonds using DTT or TSEP or something like this. So you break the disulfide bonds. Now we're going to alkylate them, alkylate the free sulfhydryls on the cysteines using an alkylating agent. And so this is to prevent well, to prevent the protein from potentially refolding and then making these erroneous disulfide bonds or having disulfide shuffling that will complicate the downstream analysis. So you reduce the cysteines, you block the cysteines, and then you add in a peptidase of interest to digest your protein into peptides. And the most common one is always trypsin, which cleaves after lysines and arginines. But depending on the protein sequence of interest and what regions you care about, you might benefit from using a different protease, such as chymotrypsin, which cleaves after hydrophobic amino acids, especially for membrane proteins whose transmembrane parts will be very rich in hydrophobics. And you will probably not get very good coverage just digesting with trypsin in these cases. Okay, but then after digestion, now you have these peptides, and then it goes through sort of the same workflow that I described in the last slide, which is can go through the MS1 here. And then if you do not fragment, then it's just considered MS1. But if you're fragmenting and then collecting data on the fragmented ions, that's called MS2 level because you're performing it again. And so I'm going to show you now what an MS2 spectrum looks like. So this is a peptide that has been fragmented by the inert gas. And I told you that this fragmentation is kind of random, but let's just look at the sequence of a peptide here. And the only things that you need to know really are that the most common types of ions formed during this gas bombardment are B and Y ions. And the nomenclature is that if the peptide, or if the peptide has an intact N terminus, then it's a B ion, and if it's an intact C terminus, then it's a Y ion. And so you can see that B2 here now would basically be methionine, and then B3 now is going to be phenylalanine. And you can see that the difference between each of these peaks is basically corresponds to the mass of the amino acid, plus or minus some other water losses which can happen. But so from this now you can identify exactly what that peptide sequence is. And so this is really, really, really important in proteomics, where you're going to have a ton of peptides, and identifying them properly will be essential. And in this case, this was other work from Shafkat, which basically they wanted to show that this phosphorylation was happening on this specific serine 205 here. And by showing this B and Y ion series and the sequence of interest, that's enough to satisfy reviewers for these kinds of experiments. Okay, just a quick, simple example about tandem mass spec and about digesting proteins into peptides. Our lab was trying to solve the crystal structure of a recombinant protein, but it would not phase correctly. And so one technique is to incorporate an unnatural amino acid into the protein that has an iodine group here, which basically helps with the structure solving down the line. But just to ask the question, okay, we produced this protein, but did the unnatural amino acid incorporate in the right place? And is it incorporated? You can take the two proteins, digest them, and then when you run them on the mass spec, this is a chromatogram here, but it's still the same, it still gets the same point across. In the wild type protein, you can see that there's a peptide here that has the this tyrosine right here, and then the tyrosine was changed to an iodophenylalanine, and there's a peptide that's unique to the mutant right here. And so this was a good quality control check for a single protein digested into peptides. And so for something like this, you only need a few micrograms as well, so it's really efficient. Okay, so now some workflow considerations for proteomics. If we're doing a single protein digest, as I showed you in the last slide or in the phosphorylation example, you really don't need much. You can get away with a couple micrograms. Now, if you're doing more complex proteomics with mixtures and cell lysates, it's always better to start with more, but you still can get good results anywhere in the microgram range. So the basic workflow for digestion, if we're doing it in solution, is you just lyse your cells with your lysis buffer of interest with some detergent to extract membrane proteins, things like this. But you're going to add urea in to denature these proteins. You might, depending on the sample prep, you might want to precipitate your protein to get rid of some of these detergents or things that might interfere with downstream analysis. Then you reduce, as I said before, with DTT or TSEP, alkylate, digest. And then after you have all of these digested peptides, the world is kind of your oyster, and you can enrich for certain kinds of peptides. So titanium dioxide or iron NTA are really common to enrich for phosphorylated peptides. And then, yeah, just sort of a procedural thing, but it's nice to desalt your peptides before running them. Digest can be either in solution, as I said above, or in gel, which I'll talk to you briefly about. And then here now, the LC gradients are going to be longer from the intact protein gradients, because we really want to separate these peptides by time so that the mass spec has a chance to identify and quantify them accurately. And so here we're just going to 40% acetonitrile because we're now working with these peptide fragments, which we'll elute earlier. And of course, you can do 60-minute gradients, 90-minute gradients, up to 3-hour gradients. Longer the gradients, the better the resolution. But of course, there's always the issue of machine time. So that's the in-solution digest. And on the other hand, we have what's known as the in-gel digestion, which can be very handy. A few advantages is that you don't have to worry about removing contaminants, such as detergents. And there's no worries about protein precipitation and resolubilizing after. Another good thing is that you can run the gel longer, stain it, and then you can cut out a very specific band of interest. And that's just not possible when you do it in solution. And some facilities will only do in-gel digestions because when you're getting samples from a bunch of different places and you're not sure what's in it, the gel really cleans it up. And so you can either resolve them and cut a band, or you can just run them right into the stacking or right after the stacking and then cut them as a single band. The only disadvantage is that it takes a long time to do. That's so if you have 20 samples, I wouldn't recommend doing it unless you can outsource it or finding an undergrad to do it for you. Just kidding. Okay, so now we'll talk a bit about when after you run your mixtures on the mass spec and you want to get into quantifying things, how do you go about doing that? And so today I'm going to focus mostly on the simplest form of quantification, which is called label-free quantification. And so there's no chemical modification or metabolic labeling. It's basically you have 2 samples, and you should have them in replicates in N of 3, and it can be, drug treatment, it could be an IP and then a control IP, something like this. And then with label-free, you basically run 1 sample at a time, and then based on the intensities of the peptides, there's some nice softwares that can spit out relative quantification values. And then you're able to average them over the runs and then create some figures and see what's enriched or what is depleted across these samples. Metabolic labeling is really, really exciting because now you can have these 2 states that one of them has a heavy isotope label that's been metabolically incorporated. So all proteins in state B will, the most common label is heavy lysine or heavy arginine. And so you can actually mix your proteins and then digest them at the same time. And in one run, you'll have a readout for the same protein, the same peptide, the light version and the heavy version. And so this is great because it allows you to multiplex your samples a bit. So you don't have to run like every single sample by itself, but it's also really good for more sensitive quantification. Okay, so I've talked to you guys a bit about the prepping of the biological samples and the LCMS, but then when it comes to actually analyzing your data from the peptide level, how does it work? And just really quickly, pretty much all of the softwares work by you give it a FASTA file, you tell the program what enzyme you use to digest the proteins, and then it'll generate all of the theoretical peptides from that FASTA file and that enzyme. And then it will go and match your experimental spectra to the theoretical peptide spectra. And then there's some statistics and scoring to see if it's a real hit or not. This is a quick tour of the software that I'm going to potentially breeze through because I'm a little bit short on time. MaxQuant is freely available, and it seems maybe a bit daunting, but it's really, really, really simple. All you do is you load in the 2 folders into MaxQuant using these buttons up here. All you have to do is name them using an experiment name. And then, as I've said, you pick your enzyme that you use to digest. And so we use trypsin, cuts after lysines and arginines. And then you can set the number of missed cleavages because it can happen and it will happen. Modifications, you can look for specific variable modifications, meaning the software will look for the unmodified peptide and the modified peptide. For fixed modifications, we have this carbamidomethyl, which comes from the alkylation of the free cysteines. Then, yeah, you give it your FASTA file and you start the run. And at the end of the MaxQuant run, it will take all of these peptides and then summarize them on the protein level. So it will assign each peptide to a protein. And then it will give you an LFQ value, a label-free quantification value, which is basically the abundance of the protein that's been normalized across samples and within samples as well. So if you're looking at protein level abundances, it's sufficient just to take this LFQ value and compare and average it and then compare it across runs. And so this was an IP experiment that got sent to us. And we had a control IP and then one that actually had the overexpressed bait. And you can see that the overexpressed bait in the IP was significantly enriched. And so I said you can just take them and average them. There's a few other transformations you have to do, but if you have questions, you can ask me. Okay, I won't get into the biology too much, but this, I just want to make everybody aware of what a volcano plot is really quickly, because we were looking for proteins that were enriched in a certain kind of mitochondrial vesicle compared to mitochondria. And so if we're looking at the volcano plot, you have the fold change on the x-axis in the log2 scale. And so proteins that were enriched in these mitochondrial derived vesicles over mitochondria, you can see on the right side here. And then on the left side means that they were depleted. So there was more in the mitochondria. And then on the y-axis is just your significance. And so negative log 10 of your p-value. And so the larger this value, the lower the p-value is. And a p of 0.05 is 1.3. And so I have a link at the end if you guys are interested in making very easy volcano plots, but this is a really common way of visualizing mass spec data. Okay, so this is now probably one of the more interesting examples for all of the pharmacologists in the audience, or all of the true biologists, because you can do unbiased screens of affinity purified samples, so of IPs or things like this, and then instead of running a million Westerns to see if if other proteins of interest came down in the IP, you can just digest your samples and then run them on the mass spec, and you get an unbiased look at everything in the mixture. And so one really cool protein from the paper from the Krogan lab is they took all of these SARS-CoV-2 proteins, put them in strep tag vectors, expressed them in hex cells, and then did affinity purifications. And excuse me, they did label-free quantification, as I described earlier. They scored the interactions using these two softwares, which are relatively straightforward, but basically just will look at your treatment and control and then give you some stats on what's truly enriched or not. And from this, they were able to create a map of what all of these SARS-CoV-2 proteins were binding to in human cells, and made some really nice bioinformatics figures here for the SARS-CoV-2 proteins, and then its human hits and drug targets, and then of course drugs that could be used there. One of the limitations of affinity purification mass spec is that it's hard to capture weak or transient interactions, and it's not the best for membrane proteins, unfortunately. Now, sort of another way of looking at interactomes that doesn't suffer from this limitation of not capturing transient interactions is the technique that's called proximity ligation. And so in this case, instead of tagging your protein with a strep tag and doing a pull down, now what you'll do is you'll tag your protein with a BRA tag or fusion protein, which is a biotin ligase. And after transfection, then you can add biotin. And in the presence of biotin, your labeled protein, the BRA on your protein, which is hopefully correctly localized, will go and label all proteins. It will add a biotin to the lysines of nearby proteins within 10 nanometers. And then from these biotin tag proteins, then you can pull away these biotinylated proteins using streptavidin beads, and then sort of do the same sort of workflow, digestion, reduction, alkylation, digestion, and then you run them on the mass spec, and then you look for, it's really important to have a good control in this case, because you want to have a background population of sort of non-specific biotinylated proteins, so that when you compare it to your overexpressed protein of interest, that you're really seeing real hits. There's a few limitations of this Bioid setup, and it's that it does label lysines, so it does rely on having accessible lysines on proteins. And for the first iteration of Bioid, the labeling times are long, but now they've made TurboID and Apex too, which operate on similar principles, but can be done a lot faster, as opposed to incubating your cells with biotin forever. I won't get into this. This was a cool finding from a Bioid experiment, but I just want to highlight this website at the bottom here, which is from Anne-Claude Gingras, and it is a public repository for Bioid data. So you can go in and search for your protein of interest, and you can go and look at its interactors all in one place. And that, yeah, it's exciting to me, but I will skip that in to allow time for questions. Okay, the last sort of technique that I'll bring up is to gain structural information from complex mixtures. And I hope that I've expressed that you can measure pretty much anything with mass spec, and if you can have a cell lysate, you can really do anything to it and then try to compare differences later on. And so to gain some structural information of not just in the proximity case of what is close to what, but now this is another layer of resolution where if we use a cross-linker that has specific reactivities, we will gain hopefully either inter-protein or intra-protein cross-links that now will tell us, okay, not only were these proteins close, but this specific peptide or this specific region of the protein on protein A was in proximity or was interacting with protein B. And so there are really nice softwares for selecting for these cross-link peptides, but it is difficult to enrich for them. I will just say that. So it's important to there are a few cross-linkers that have cool things like biotin moieties in them that you can do a similar enrichment like in the last step. But then basically after searching, then you can now, hopefully with a technique like this, gain an idea of where proteins are actually binding in an unbiased way. And you can do this in a whole cell lysate. There's one last technique where they shoot lasers at things that I won't get into, but I've now compiled a few of the resources that I found quite helpful over my time in trying to teach myself a lot of this. And I think that the slides will be distributed, which is nice, but if not, you can screenshot it or you can email me. But yeah, so there's a facility, just to say briefly, if you are interested in doing any intact protein analysis, please reach out to me, Mark Hancock, or JF, because we have this set up in the McIntyre. For proteomics, we will probably suggest that you outsource this either to the MUHC or to potentially the platform manager in McGill Biochem. And then, yeah, there are some data repositories and some links for some easy tools for data manipulation. And with that, thank you to everybody, and thank you to you guys for listening for so long.
Awesome. Thank you so much, Andrew. That was super informative. So we have some time left, like 5-ish minutes, for any questions that anyone in the audience may have. And just a quick reminder to please check your little DMs in case you're one of the winners of the Uber Eats gift cards. So does anyone have any questions? I guess you were just too clear, Andrew.
Yeah, or too confusing. Everybody's brain has turned to mush. Hopefully the former.
I guess I have a question that's just out of curiosity. Was there anything like while you were learning how to do mass spec and all of that that really surprised you, or things that like maybe based on the theoretical knowledge you learned from class was actually very different in practice?
I'm constantly surprised with everything in the mass spec. When you expect things to work, things don't like to work. But I'll say that I think I kind of jumped into it really from a practical side first, and then the theory started to come after, and only after like years of processing samples and then piecing it together bit by bit. But yeah, there's always surprises. It's funny when you have a protein that should show up and then it just doesn't ionize, and then you just go, well, yeah.
Okay, cool. It looks like Yuri and Braden both have questions also. Maybe Yuri, you can go ahead.
Okay, yeah, thanks. Yeah, very clear presentation actually. So I'm curious about the sample workflow for the mitochondrial vesicles that you're purifying. So you ultracentrifuge the vesicles and then just do plain extraction and run it on the mass spec. Is that it?
Yeah. So we did ultracentrifugation, and then we had a Perkel gradient, and then we took this really specific interface that between the 40 and 50% region, and then from that we just did an in solution digest. And one issue with that project was that the amount of MDVs in that gradient was super low. So we were operating less than a microgram of protein in the like maybe 100, 200 of nanograms of protein. So JF is squinting his face there, but
so is that enough for discovery? I mean, a priori, you know more or less which proteins might be there, so you're looking. But if you're going in a completely unbiased discovery phase, what are the limitations?
Yeah, it is enough for unbiased discovery, definitely, especially with some of these newer instruments. Like on our instrument, we were getting maybe 40 hits, that like 40 to 50 protein hits. And then we sent it out to one of these more cutting edge machines that has ion mobility built in. And then from there, we were in the hundreds and hundreds of hits. And so from the nanogram range, the real issue is minimizing any sort of sample handling, because every step you lose some. But if you can have in the hundreds of nanograms, you can still do unbiased identifications on the new end machines.
Okay, and last quick comment. When you don't see an ion, do you look on the? You keep talking about the positive ionization, so you look in the negative phase or mode. Routine.
I guess you could. I've never done it, but for me, I've always chalked up negative mode to doing like oligonucleotide stuff. But when that happens, we usually go to the MALDI first and then put the protein on a different ionization source. And in the cases where we haven't seen it on our electrospray in positive mode, we've always seen it on MALDI.
Okay, yeah, thanks.
Awesome, thanks. Braden, do you still have a question?
I do, yeah. First and foremost, wonderful presentation. I had a question a little bit when you were talking about like some of like the label-free quantification versus some of the other different styles that you could potentially do. I was wondering for that kind of like light versus heavy metabolic one where you can kind of do some of that multiplex. So I guess I kind of have a two-part question. So the first is, how do you basically get like the light or the heavy proteins incorporated into the cell? Is it just a supplement that you add within the media when you're growing them and they take it up during like cell division? Or is there a more complicated process to that?
You buy heavy labeled media and then you just incubate your cells with them. And you can even do that, like we did that with mice by feeding them heavy leucine chow. So I mean, you have to wait a bit longer for it to get incorporated, but after some weeks on the food, then yeah, it gets, same premise.
Okay, fantastic. And so the other question I had is, I think you had one that was kind of in the middle that said like TMT, which I think was a little bit different than the two of them. And I was wondering if you might be able to explain that one a little bit.
Yeah, that's the most powerful one of all. That is, you chemically label the peptides using mass reporter tags that have, so basically you can, in one run, let's say you have like treatment 1, 2, 3, and then control 1, 2, 3. You label each of the 6 with a different mass reporter. So you'll have 6 different things. And then you can combine those peptides into one run. And on the high-end mass spectrometers, you can then look at the quantification of all 6 samples in one run, basically based off of the mass reporter, which has it has a specific fragmentation pattern. So that's very sensitive, better than LFQ. It's expensive, but it allows you to multiplex really well. And that's sort of like the gold standard now. Yeah.
Cool. All right. Thank you so much.
I think Dan has a question.
Yeah, for sure. Go ahead.
Sorry, I tried to use a culturally sensitive hand color, but it fades in the background. So I have 2 questions. First of all, great job. First one's quite specific. Second one, I guess, is a little bit more general. So you talked about the in-gel analysis, and you said you didn't have to worry about detergents, but there's SDS there, and I just wonder what consideration you have to give to that.
Yeah. You don't have to give any consideration because all the steps are the same after you run it. You excise the band, and then you will draw out the liquid with acetonitrile and then reduce alkylate, blah, 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 blah. But at the very end, you have a band, or you have a diced band that has gone through the digestion process where the trypsin migrates into the gel. And then the last step is extracting the peptides with acetonitrile. And so you do a couple of extractions with pure acetonitrile or with a high percentage of acetonitrile, and then the SDS is not a big issue at that stage.
Okay. And then I guess a sort of more general one in terms of newer methodologies, and I know this will, maybe JF will want to end up answering this, but for single-celled proteomics, especially in light of what you were just discussing with Uri about how much protein you need. Like, is it done by mass spec? And if so, like, how do you enrich for really low abundance proteins?
This for single cell proteomics?
Yeah.
I'm tagging in JF on this one. JF, it's your, it's.
I know, it's my turn. Yeah, it is. I mean, so proteomics just means that you're quantifying proteins at the same time. It doesn't have to be mass spec based. But of course, if you want to tag and do it in a non-biased fashion, then it has to be done by mass spectrometry. And yes, so the newer instruments, they do it using the same kind of instrument that Andrew has been talking about. They're just better. They have better optics, faster components. They do all kinds of tricks that I won't go into to increase the sensitivity. But the trick is also upstream of this. So I think, so Andrew alluded to this, right? That one of the issues is sample prep. So proteins are sticky. So as you stick your pipette tip into a solution in and out and in and out, there's more protein getting absorbed on the surface. And then so if you start with a cell, so a HeLa cell, I think is about 0.5 nanogram of material. And there's nothing left at the end, right? So what people have devised is things called one-pot processing methods, where you get your cell into like, a 384L well, and then you basically throw reagents at it using piezoacoustic liquid delivery, right? So you just throw hundreds of nanoliter of your trypsin, and then you acidify it, and all is done in one pot. So because it's in one pot, you have less loss through surface adsorption. Yeah. So but yeah, people are doing this now.
No, I know. That's why I was curious to learn a little bit more about.
Another trick that refers to one of the things that Andrew talked about is the isobaric labels, that these TMT labels. So what you can do is actually have one channel, which is actually a bulk. So you can take 100 cells, pull them. That's going to be your channel 1. And then every other channel is single cell. So you're using what we call the carrier channel, right? So because everything is pulled together, and there's a huge dynamic range for these TMT labels, then you can take advantage of having your bulk sample pooled with all your single cells. So you can quantify each of those cells compared to your pooled sample.
Interesting. Great. Thank you both. Good slides, Andrew. I think you're going to get roped into teaching that in a class at some point.
I am enrolling Andrew for 505 next fall, actually. Yeah, very good job, Andrew. That was fantastic.
For a small price, and this is being recorded here, so.
So what does that mean? If you get run over by a bus, we can just play the recording, or?
Yeah, just on
Awesome. Thank you so much, everyone, for attending, and thank you again, Andrew, for agreeing to give this wonderful presentation. I hope everyone learned something today. I think this is the last of the technical seminar series that will be happening until fall semester, so keep your eyes out for the next email that will come all the way in September. But thanks again, everyone, and I hope you have a great rest of your day.